Good afternoon, everyone. My name is Paul Musey, and I'm a clinician researcher at Indiana University, and I want to thank you so much for joining us for the SAEM21 National Grand Rounds Senior Faculty Research Highlights. Our goal in this session is to highlight high-quality research and high-impact research results from some of the most established clinical investigators within our field. And thus, this session will feature two dynamic presentations from Drs. Carlos Camargo and Dr. Lance Becker. Our format will be as follows. Each of our special guest presenters will present on their chosen topic for 20 minutes each, after which, depending upon the size of our group, we will either have one breakout session for Q&A and networking or split you all into breakout rooms to allow you to interact with our speakers for the QA and networking session. After 20 minutes, we would switch presenters so that you would have the opportunity to interact with both. But really, you all didn't join to hear from me, so let me introduce our first speaker. Dr. Carlos Camargo is a professor of emergency medicine as well as epidemiology at Harvard University, as well as the Kahn Chair in Emergency Medicine at MGH. He founded and leads the Emergency Medicine Network, EMNet, an international research collaboration with 247 hospitals. Major areas of EMNet research are the management of respiratory and allergy emergencies, the etiology of childhood asthma, and as well as allergy, and health services research on emergency care, such as the National ED inventory and workforce issues. His research also focuses on nutrition and respiratory and allergy disorders, both in large cohort studies, for example, the nurses' health studies, and in large randomized clinical controlled trials. Dr. Camargo is past president of the American College of Epidemiology and has worked on several US guidelines, including those on diet, asthma, and food allergy. In 2016, he received the American College of Emergency Physicians Award for Outstanding Contribution to Research. He has over 1,100 publications and has an H index of greater than 141. So without further ado, let me introduce Dr. Carlos Camargo. Thank you.
Good afternoon. My name is Carlos Camargo, and thank you for the opportunity to speak at the National Grand Rounds. I'll be talking on the heterogeneity of infant bronchiolitis. I'll start with an introduction to the Emergency Medicine Network, or EMNet, and then transition to the focus of this talk, which is bronchiolitis. Specifically, I'll be comparing respiratory syncytial virus versus rhinovirus differences in a variety of omics data, and then using latent class analysis to identify severe bronchiolitis profiles. And finally, to talk about integrative omics and its role in defining endotypes of severe bronchiolitis. I'll summarize and discuss a little bit about next steps. The Emergency Medicine Network was founded in 1996. And its mission is to advance public health objectives through diverse projects in emergency care, particularly multi-center clinical research. There are a variety of projects and acronyms in this network. Funding is predominantly from the government. And at this point, we have more than 800 publications over these past 25 years in all age groups and diverse topics. The leadership of EMNet is shown here and includes several faculty members, Dr. Cash, Dr. Hasegawa, Dr. Samuels-Callow, Dr. Zackerson, and Dr. Zhu, and also Ashley Sullivan, who is the director of the EMNet Coordinating Center. There are EMNet sites across the USA and even some overseas. They're shown here, including with the red circle, the Coordinating Center, which is based at Mass General Hospital in Boston. EMNet research focuses on three areas: respiratory allergy emergencies, such as asthma, anaphylaxis, bronchiolitis, and these fall sometimes under the label of MARC, or the Multi-Center Area Research Collaboration. We also perform health services research in emergency care and do other public health projects. Shifting now to the focus of today's talk, let's talk about bronchiolitis. And I put it in quotes because it's a clinical syndrome with multiple definitions worldwide. most agree that it's an acute lower respiratory infection with inflammation caused by one or more respiratory viruses, typically respiratory syncytial virus or RSV. The American Academy of Pediatrics includes all children aged less than 2 years, but this is controversial internationally. There are concerns about overlap with recurrent virus-induced wheezing, overlap with virus-induced asthma exacerbation, and for that reason, some people limit bronchiolitis to age less than 1 year, others only to the first episode, others limit it to RSV only. Bronchiolitis is important for many reasons, but 2 are listed here. It's acute morbidity. It happens to be the #1 cause of infant hospitalization in the USA, with about 130,000 hospitalizations each year. And this affects about 2 to 3% of all infants. It also has chronic morbidity. Among hospitalized infants with bronchiolitis, or what I'll call severe bronchiolitis from here on, about 30 to 40% will develop childhood asthma, which means that most will not. And it's not clear why. You may be familiar with the American Academy of Pediatrics guidelines for bronchiolitis, which are most notable for the lack of any recommended testing, treatment, or vaccine. So the most common reason for infant hospitalization has none of these options, which is a puzzling situation and one that we have been focusing on for the past few years. Shown here is our theoretical model of severe bronchiolitis and later risk of childhood asthma. Key points are that RSV is indeed the most common cause of bronchiolitis, but other viruses are also involved. The second most common is rhinovirus, sometimes abbreviated as RV. And this affects about 10 to 30% of severe bronchiolitis. We believe there's an interaction between these viruses, the microbiome in the child, the host response, and the overall environment to lead to different types of bronchiolitis, which in turn lead to different types of asthma. Over the years, we've created several infant cohorts, which are shown here, the Mark 30 study, the Mark 30 Finland study, Mark 35, and Mark 43. And going back to that acute versus chronic distinction, these different cohorts address different elements. For instance, the Mark 30 USA was focused on severe bronchiolitis from the emergency department to the inpatient setting, but didn't really have a chronic follow-up. So asthma was not something we could study with that NIH-funded project. In Finland, however, the same protocol was used, and because of Finnish national data, we are able to look at asthma status 6-7 years later in the children who are enrolled in that study. In Mark 35, which is ongoing, we have combined the best of these earlier studies, and so we not only have information about the acute event, but also the long-term follow-up. In Mark 43, we are looking at healthy infants as a counterpart to the Mark 35 study, and they're also being followed to school age and using the same techniques, the same approach, same data. So it provides a very nice approach to compare the Mark 35 data with what one sees in healthy children. To briefly summarize the Mark 35 study, also known as the Wheezing Index or WIN study. The study population are infants aged less than 1 year who have been hospitalized for bronchiolitis. We initially enrolled 1,016 kids at 17 sites over 3 fall-winter seasons. And among this group, 921 are being followed for long-term outcomes. Data collection involves interviews every 6 months, medical record review by clinicians every year, and then in-person exams, not only at enrollment, but also at age 3 years and 6 years. We've collected a variety of early life biospecimens, including blood and index nasopharyngeal aspirate, multiple nasal swabs over time. And the main outcomes of Mark 35 are age 3-year recurrent wheezing, age 5-year doctor-diagnosed asthma, and age 6-year asthma phenotypes. Shown here is a quick comparison of the children enrolled in the Mark 35 versus Mark 43 cohorts. In MARC 35, the average age of enrollment, which again was a severe bronchiolitis hospitalization, was around 3 months. And 60% of the kids were boys, which is what one would expect to see. And about 50% of the enrolled children are non-Hispanic Black or Hispanic. Interestingly, about a third of these kids had parents who reported asthma. In MARC 43, we tried to enroll around the same age. We got kids enrolled at around 4 months. Again, about half of the population is non-Hispanic Black or Hispanic. And it turns out that about 30%, 29% of the parents also reported asthma. The initial enrollment was in 14 states, but over time, these kids their families have spread out all over. And you can see here that the participants now are in many states across the US and even overseas. So with that background, let's now transition to the heterogeneity idea that, for instance, RSV versus RV bronchiolitis are different in fundamental ways. This work was done by Jonathan Mansbach from Boston Children's Hospital, and it examined the nasopharyngeal microbiota by virus. And what you see here is that if you look at specific bacteria, for instance, Moraxella and Haemophilus, that the RSV group had less than the RV group. The opposite was true for Streptococcus in the airway. And it's very important, but these findings were replicated in a separate cohort with similar biospecimens and analysis. So these are pretty clear evidence that as one approaches a child with bronchiolitis, that the airway bacteria, which could play a role in a variety of both acute and chronic outcomes, are different in those children with RSV versus RV bronchiolitis. We next looked at the nasopharyngeal metabolites and whether they differed by virus. And what you see here is a very clear separation between the metabolites involved in RSV bronchiolitis and rhinovirus bronchiolitis. The differences were highly significant. Looking more closely at the specific metabolites, you can see a variety of potential targets for therapy or simply for a better understanding of the pathophysiology of severe bronchiolitis by virus type. In a subsequent paper, we looked at the serum metabolome and performed a similar analysis. And while the split is not as clear, again, we do see differences in the serum metabolome by virus type. There were enriched metabolites, for instance, in lipids and amino acids that differed by virus. We next investigated nasal microRNA profiles and whether they also differed by virus. This work was led by Kohei Hasegawa and with testing and input from Rob Freistadt at Children's National Medical Center in Washington, D.C. And again, you see pretty clear differences between RSV bronchiolitis and RV bronchiolitis using different approaches to this data analysis. We also were interested in host response and how that played into this. And we looked closely at a paper from 2012, which discussed the role of CCL5 or RANTES on risk of asthma-related outcomes. These data come from the REBEL cohort, which is 206 children with RSV bronchiolitis only. They were enrolled at hospitalization, and about 50% of them developed asthma. And what the investigators found was that higher CCL5 in nasal epithelium was a strong predictor of asthma at age 6 years. In the MARC 35 cohort, which involved 921 infants with any infecting virus, we found our overall results were null. There was no apparent association between nasopharyngeal CCL5 and risk of recurrent wheeze at age 3 years. However, when we split out the children with RSV-only bronchiolitis to match the design of the REBEL cohort, we then saw that the higher CCL5 levels were indeed associated with higher risk of recurrent wheeze. And that also held true for asthma. So what we're seeing here is that if you don't take into account the inciting virus, you will see different results. Again, strong evidence to support the heterogeneity of this condition called bronchiolitis. So shifting gears, we wanted to use a different approach to understand the heterogeneity beyond just RSV, yes, no. And we did this using a technique called latent class analysis. And this project was led by Orion Dumas, who is now working at INSERM in Paris, who was a postdoc in my lab. And we noted that prior studies of potential bronchiolitis heterogeneity had classified children according to a limited number of characteristics, like RSV yes, no, which is what I've been showing you for the last few slides, or in a single dimension, like very severe or not so severe. And we thought, let's let the data tell us what groups are present, and then examine the trajectory of those kids as identified by the data. Starting with the MARC30 USA cohort, we identified 4 profiles, which are shown here. And the first is, I think, the most interesting. We call it Profile A, and this is predominantly rhinovirus bronchiolitis, children with a history of wheezing, history of eczema, who are wheezing at presentation to the emergency department. And it's only about 12% of the kids, but we suspect that these are children with early asthma. Profile B sounds a lot like classic bronchiolitis, with the predominance of RSV and the other characteristics shown. Profile C is, again, very heavy RSV, but also very sick. And then lastly is Profile D, which was an odd group, sort of atypical and more mild. And what's important here is that we replicated these basic profiles in an entirely separate cohort called MARC30 Finland, which I presented to you earlier. And so we believe that these profiles are real and that they speak, again, to the heterogeneity of bronchiolitis. We use a similar analytic approach to look at these profiles in the MARC35 cohort. And again, we found Profile A, about 15% of this group, Profile B, very similar, predominantly RSV, and then Profile C, again, predominantly RSV and sicker. Interestingly, in MARC35, we do have biospecimens, and so we could look, and what we found in Profile A were higher blood eosinophil counts, cathelicidin levels, and higher proportions of Haemophilus or Moraxella dominant microbiota. When we looked at the trajectory of these kids over the years that we've been following them, we see a very clear increased risk of recurrent wheeze for Profile A, intermediate for Profile C, and then Profile B, the classic RSV bronchiolitis, is the baseline here. And you can see has the lowest risk of developing recurrent wheeze by age 3 years. In terms of asthma, we also were able to look at ICS or Montelukast use at age 3 years. And we also look at doctor-diagnosed asthma. And again, very consistent findings, where Profile A has the highest risk of developing asthma. In subsequent analysis, we've extended this, and again, Profile A is a group that is different from classic bronchiolitis caused by RSV, and again, speaks to the heterogeneity of this severe bronchiolitis presentation. With regard to next steps within the MARC35 and MARC43 cohorts, we currently are expanding the metabolome and microRNA testing to the entire MARC35 cohort with funding from 2 RL1s. We recently completed a proteomics pilot study with an RL1 admin supplement. We recently got an RL1 to look at genetics and epigenetic factors in the 2 cohorts. So this involves not only genotyping, but methylation studies. And just to close here, we're doing ongoing systems biology work to integrate all of this multi-omics data to identify endotypes for both severe bronchiolitis and asthma. Just a few words on endotyping using multi-omics data. As shown here in this graphic, on the left side you see a very heterogeneous group of people. And what we are trying to do here is use unsupervised machine learning methods to identify biologically and clinically meaningful endotypes. In other words, using all these different omics data and also environmental data and sort of more traditional clinical data, can we divide up the bronchiolitis population into endotype A, endotype B, endotype C, identify the high-risk endotype, and then use that information, for instance, to guide acute therapy or to guide prevention strategies in high-risk groups. The search for bronchiolitis endotypes is led by Yoshireta and Kohei Hasegawa. And right now we're focusing on the data that we have, which is soon to expand enormously, as I mentioned earlier. But using just clinical data, airway microbiome, and host response, we've examined 221 of the MARC35 infants with RSV bronchiolitis and found endotypes that differ in terms of their risk of developing asthma. This was a plenary presentation earlier today at SAEM. We've also looked at 122 infants with rhinovirus bronchiolitis using similar techniques, and again, we're able to identify an endotype that had higher odds of asthma. So in summary, I submit to you that bronchiolitis is not only common, but it's heterogeneous. And focusing just on the virus type reveals clear difference in airway microbiota, metabolome, and microRNA. We saw clear differences in the role of CCL5 on risk of recurrent wheeze at age 35 years. The latent class analysis reveals reproducible profiles with difference in biomarkers and in risk of multiple asthma-related outcomes at age 3 years. And most recently, the integrative omics analyses that we're doing are revealing bronchiolitis endotypes, which have implications for clinical outcomes. With regard to the treatment of severe bronchiolitis, I think this heterogeneity argument argues against a one-size-fits-all recommendation. We strongly encourage improved characterization of severe bronchiolitis type in future research, especially RCTs of therapeutics. For instance, bronchodilators, corticosteroids, montelukast. Would these asthma treatments work in a subgroup of bronchiolitis? Perhaps. So rather than wiping them off the map, perhaps we should look at subgroups in which it's more likely that they would work, and then we might have some targeted therapies for this very common presentation of severe bronchiolitis. Likewise, with regard to the chronic morbidity and the potential for primary prevention of childhood asthma, we're identifying a variety of potential targets which we could limit or block, or if they were seemingly protective, we could enhance or give to try to prevent childhood asthma. Finally, I want to acknowledge the many people who have contributed to the work I've shown to you today, including the site PIs and locations of the MARC35 and MARC43 sites, and also to list some of the NIH funding that has been used for the research I've shown to you today. Thank you very much.
It is my pleasure to introduce our second guest speaker, Dr. Lance Becker. Dr. Becker is also an international giant within our field and is professor and chair of emergency medicine at North Shore University Hospital and Long Island Jewish Medical Center, as well as chair of emergency medicine at the Donald and Barbara Zucker School of Medicine at Hofstra, Northwell. He has translational research interests that extend across the basic science laboratory into animal models of resuscitation and to human therapies. He has been internationally recognized as a leader in the field of resuscitation for more than 25 years and has pioneered advances in improving the quality of CPR, AED use, and defining the three-phase model for cardiac arrest care and therapeutic hypothermia. He has received numerous awards and honors for his work from the American Heart Association and the American Society of Critical Care, just to name a few. Dr. Becker is also an elected member of the Institute of Medicine and National Academy of Medicine, and has been a leader within these organizations' efforts to improve survival from cardiac arrest and help write the recent strategies to improve survival from cardiac arrest, A Time to Act. and is currently working on developing a national collaborative to improve survival from cardiac arrest. It is my pleasure to introduce to you Dr. Lance Becker. Thank you.
Hello, everyone. My name is Lance Becker, and it's my absolute pleasure to have a little while to share some ideas with you on resuscitation science discoveries and the really bright future that I see for emergency medicine in the coming years. I put a note to remind myself that this is really, I think, one of the biggest times of opportunity that emergency medicine has ever had, and I hope I can share some examples with you about why I believe that's true. These are my disclosure information. You'll notice I do have grants and sit on some science advisory boards. I think the most important thing to note is that I have an intellectual conflict of interest, if you will, or a bias. And that bias is that death is far more reversible than we currently believe. And the corollary of that is we allow many people to die prematurely. You will see that that is a bias that really does extend through many of my comments. And so I'm going to own that in a transparent way. I want to give a shout out of thanks to my laboratory at Northwell Health that has supported me. You'll see some of the members there. I can't call them all out, but whenever somebody gives the kind of talk that I intend to give, you know that there are just a whole lot of people who have helped them with all of the work that's being done. Really, none of it have I done by myself. So in the next few minutes, I want to tell you a little bit about me because most of you don't really know me, my career path, a few of my successes. And in addition to successes, I think one thing we don't talk about enough are the things that we have not yet been successful with, sort of our failures, if you will, or just things that we haven't managed to accomplish in our careers. And I want to highlight again what a bright future, what a great time this is to go into emergency medicine, and a little bit about in the resuscitation space, where I see emergency medicine's future. Now, I'm going to give you sort of a bottom line takeaway that I hope you will take away a little bit from some of the examples of my career, what I've done, which is that I believe the true test of an academic physician is not about your successes. Yeah, I get to give this talk because I've had a lot of successes, but it's really rather what you do when you do not succeed. So to those of you who are starting your career, I suggest be bold, take chances, but expect failures, and then be persistent. Go after those things that didn't work. And every now and then, of course, you get to celebrate your successes. Now, about my career, for those of you who don't know me, I started at the University of Chicago, and I spent many years there, and then I got recruited to the University of Pennsylvania and spent between those two jobs, that was about 25 years, and then about 6 years ago, I transitioned to become the chair of emergency medicine for the Northwell Health Organization. And for those of you who don't know anything about it, because it's a relatively new organization, very large health system in New York, does 30% of the healthcare for the state, 19 emergency departments, there's 3 emergency medicine residencies, and over 900,000 visits. And in addition to the operational kinds of things, I run a critical care laboratory that is focused on resuscitation science. So that's kind of, if you will, in a nutshell, my career arc. Now, in addition to sort of those organizations, there were other organizations that really helped my career, and I want to just thank them and highlight them, like SAM and ACEP, the American Heart Association, that was kind of my home for a lot of the things that I did in cardiovascular care, NIH, the national academies, and of course industry partners that have, many of them I've worked with some of the industries for 20 years as well, and they have been critical to the successes that I've had. In addition, when you are really embedded in science the way I have spent a lot of my career, you develop your own just sort of resuscitation science community of scientists who keep in touch. And I feel very, very fortunate that, I communicate with people all around the world on a regular basis around the ideas of resuscitation science. And so this is kind of my career arc and the organizations that were important to me. And now I want to turn a little bit to the ideas that have been important to me. Because in fact, it was really ideas that were far more important in some ways than where I was. And the first idea that really sort of captured me was the idea of understanding a little bit more about cardiac arrest epidemiology. Who gets it? Who survives? Where are the opportunities? And that really led me into these areas of CPR quality, AEDs, hypothermia. And I'll say a little bit about that. But then the next idea that really captured me was the idea that sort of focused in cellular biology was sort of the secret, if you will, of life versus death. And I definitely spent a great deal of my career examining questions around when do cells die? Why do cells decide to die versus live? And that led me to reperfusion injury, to mechanisms of pathobiology and reperfusion injury, and to new treatments. And of course, that also led me to develop a basic science laboratory, something that I feel we have far too few of in emergency medicine. And then the next idea that really captured me was the idea of mitochondrial medicine and its connection to emergency medicine, that energy is central to life, that mitochondria have this very important dual function that is both giving us energy, but it also gives us death. And then the modulation of mitochondria and mitochondria energetics, and this is something that I think I won't be able to speak about much in this talk, but is just turning into an enormous area of fabulous interest around the world. And finally, I want to say a few bits about advanced resuscitation in emergency medicine for cardiac arrest using things like ECMO and ECPB or ECPR, and the idea of combining a pump with multiple metabolic therapies. I'll probably call this a cocktail as I go through this, where you give multiple agents to try to correct the metabolic disarrangement that takes place when someone has a cardiac arrest. And of course, it's very, very important in my view that emergency medicine has translational science laboratories. Now, just a few examples of some of the things that I've really been involved in. Early in my career, I mentioned the epidemiology. This is one of our papers on racial differences in the incidence of cardiac arrest and subsequent survival. And this is kind of a great example of the kind of things you can do when you develop new data that other people haven't been able to capture. That's what I did when I was in Chicago. I also, with Mike Weisfeld, wrote this paper on resuscitation after cardiac arrest, a 3-phase time-sensitive model. And the last time I checked, I think this paper has had over 1,000 citations. And another thing that we did was the O'Hare and Midway Airport experiment with the use of automatic external defibrillators. And so these are papers that have had some major impact. And, the first paper, really, we identified this major disparity in our care, the differences in survival between blacks and whites. And obviously that is still a very current issue for all of us in emergency medicine. And the three-phase model is still our current model of cardiac arrest. And you'll see that much of my attention, though, has moved towards the metabolic phase. And in terms of the AED study, the result of this paper is that, like within a year, most airports around the whole world put AEDs in their airports as a result of this paper that we published. And so these are great successes, and I am enormously pleased with them, but I don't want anyone to walk away with the idea that there aren't some things undone, that we weren't completely successful. So for example, in that first paper, we identified major disparities, but we did not fix it. We weren't able to get to the issues around structural racism that really underlie a lot of this. And, this is going to be part of the opportunity of emergency medicine in the future. If you look at the three-phase model, despite the success of that, it's kind of old. It was published in 2002. Science is moving on. We still haven't changed beyond that, and we still haven't found a very good way to treat the metabolic phase. And so even though we described, if you will, this model, it's not like we can treat it very well. And finally, when you think of public access defibrillation or AEDs went global, but it's crazy you can't find them when you need them. And if you do, this is just one of these nonsensical things, They have these little scooters you can rent for $2, and you can trace them down within 6 feet of one another, like these little scooters, or they're all over cities, but no one knows, and they're all on a map. You can see where every single one of them is. They have a GPS locator on them, but you can't find an AED very often to save your life, even if one is close to you. So there is so much that we have not done. And despite the successes of all of these, there is so much more work to be done, and areas where we have not yet succeeded. And I just want to take a moment and say those are the kinds of things that create opportunity for the next generation. And I hope many of you will seriously consider if there are not things that we can't do to now really fix some of these things in a more substantial way. Now I want to turn a little bit and say a little bit about what I'm really focusing on now at Northwell, and the work that we're doing there really is focused on things like ECMO, quality and implementation. I'll talk a little bit about one program there, but also things like oxygen metabolism, mitochondrial medicine, lipid metabolism, and finally this idea of cocktails. And so I'm going to just talk a little bit about two that I think are very practical things that everyone here would have the ability to be able to learn from and maybe even take back to their own hospital. So first I want to turn to quality and making CPR better. And great paper by Paul Chan. And what he really says is, look, you cannot fix what you do not measure. And so we got interested in ascertaining the quality of CPR. And we had already helped develop a device that I hope some of you are familiar with it. There are several devices that are out on the market that allow you to look at the quality of your CPR, the depth, the rate, the number of ventilations, and most particularly the hands-off time. And this was work that I did over really a decade. But when I came to Northwell, I really wanted to bring it up a notch. And we asked, what more could we do? And one of the things that we realized is we could do this. Does everybody recognize that little thing? A camera, a video camera. And so we started videotaping our actual cardiac arrests in the code room. And I want to just quickly say a few words about this study. We call it Mechanical Team-Focused Video Reviewed CPR. And Dan Ralston and Timmy Lee, who are pictured there, really the movers and shakers on this. They published it in terrific journals, and it's something that I think is very important for emergency medicine, which is to really get with improving the quality of a code. And what we do is when there's a cardiac arrest, we call a code, the camera gets turned on, we record the code, and then we do a video review. We actually do them on Thursday mornings at 8 o'clock. And then a month after those videos are reviewed, they get deleted. We do a chart review. We pull a whole database together. We bring everybody together to review these videotapes. There you can see Dan in a morning report looking at one of the videotapes, the attendings, residents, nurses, techs, everybody gets to participate. Everybody learns from it. And the amazing thing is we've really learned so much from this. We now have over 300 cardiac arrest cases that we've reviewed in this way. And we found all kinds of areas for improvements where we have pauses and lack of communication, problems with equipment, too many people in the room, too few in the room. And these are just some of the qualitative comments that people make. You know, I know we are not perfect, but we are so much better now. I can't believe how bad we were doing just a few months ago. This is from one of our nurses that came to our review. One of my attendings said, I never really thought about how hard it is to perform a good resuscitation. And you just can see that when you see things, it's so much better. And I love this comment from another one of the nurses. The whole culture is changing now. And, you might ask, does it do anything for survival? This is our survival data that is from the year 2017, 2018, 2019. And you'll see just on the right-hand side that the survival to discharges was 3.1%. That's to get out of the hospital. And you can see it's nearly tripled over 3 years with this program. So I encourage people to read some of our papers on this, but this is like a technology that you can bring to your own emergency department, and you can start improving the success of your resuscitations right away just by doing something with a videotape. Now I want to spend a few minutes on future horizons, and I'm going to talk about now sort of really advanced resuscitation. And the piece of it that I want to talk about while it's in the setting potentially of ECMO, I really want to talk about the idea of a cocktail of using multiple agents to take care of multiple metabolic problems. And I may refer to it as NWH9. Sometimes we call it love potion number 9. That's because the data that I'll show you a snippet of involves that particular iteration of our cocktail. And first, why do we need a cocktail? Is this just this is some data that we published about 2 years ago on the metabolomic alterations in human plasma after cardiac arrest. And the reason I show you this slide is that stuff that is in white is the same as it is at baseline, but anything that is either green or red means it is markedly up or down after a cardiac arrest. And this is human data. So what you can see just very quickly without, you don't have to know anything about the biochemistry except to say that there are multiple abnormalities. And so it's an illogical hope that one drug is going to fix all of this, because that just doesn't happen. So the thing we've really been working on is the idea of an anti-reperfusion injury brain-focused drug cocktail to take care of all of those metabolic alterations. And so we know that with ischemia, oxygen goes down, you don't have substrate, and rapidly your ATP runs out. Then you start developing reactive oxygen species, or ROS. You get calcium and ionic dysregulation. And in ischemia, calcium is pretty much a bad actor because it starts more things. And then you get the full catabolism, autophagy pathways, starvation pathways. The whole metabolism starts to become very, very rapidly altered. This happens within minutes of a cardiac arrest. And as it progresses, what you see is additional abnormalities. So you begin to see mitochondrial dysfunction and ROS and electron transport chain, calcium apoptosis kinds of issues. And these are all the cell death pathways simply become very, very active. You see terrible lipid membrane dysfunction, lipid oxidation, and in fact, even membrane disruption, which once the cell membrane breaks open, most people think that is a cell that can no longer be salvaged. And you see enormous calcium and ionic dysregulation. The cells become swollen and fragile, and there are a whole series of pathways, including the calpains, that become very active, and they injure both cardiac cells and brain cells. And finally, the whole energy pathways of the cell are enormously disrupted. If you look at ATP, there isn't any ATP left. And it's been degraded to ADP, to AMP, to adenosine. And there's enormous substrate depletion and terrific signaling that goes on. And then what we know is that the result of this is it calls out the cell pathways that really bring us to death. And so what do we do about this sort of very intensive kind of situation? And, our notion is that what we need to begin to do is to target specific therapies at many of these pathways. And as we target many of these pathways, and if we could target all of them, or the majority of them, we think there's a very good chance that instead of cell death, we could end up with a happy cell. And that's really been the focus of our work for the last several years. And what that looks like is in the setting of standard ACLS, everyone in this audience knows too often it fails. And so we are now coming in with veno-atrial ECMO, doing a cannulation, beginning to try to resuscitate that person. And the thing that we really want to do is add this idea of a cocktail of a multiple drug. And I'm just going to show you some very preliminary data. This is unpublished data. I'm not going to go into exactly what is in it. But the kind of effect that we see when we do this is seen in this panel on the left, where in the green line at 80% survival after 72 hours in rodents are these surviving animals when they get this cocktail. And by contrast, the injury, this is a 12-minute cardiac arrest in rodents, it is 100% lethal. So 100% of the control group dies within 72 hours. And I don't need to tell you that that's the kind of thing that, really could change the survival curves that we currently deal with. We're continuing to try to develop this, and we're making, I think, very good progress with it. It is very hard work because people don't really know how to deal with a cocktail, but this is really where I think emergency medicine is going in terms of resuscitation. And I do believe that in 5 to 10 years, everyone in emergency medicine will be using advanced resuscitation technologies that include ECMO and will include cocktails to bring patients back to life. So I just want to wrap up. Thank you all for your attention and remind you that very much for me, the most important thing in my career has been the ideas that I've had a chance to explore in emergency medicine. And I want to just leave by saying that I believe this is the greatest opportunity. We just have such a great opportunity right now in emergency medicine to make such impact in the world that I know we have been suffering with a pandemic, and we're suffering with strife and with racial injustice and with political fiascos, and there are so many things that people are emotionally wrapped up in right now, and rightfully so. But this, I think, people will look back on this decade and say this was one of the most opportune times to go into emergency medicine. I encourage everyone to sink their teeth into the things they're passionate about, to make discoveries, to do research, and to really bring the kind of discoveries that emergency medicine can bring to the house of medicine. I thank you for your attention.